Okay. Hello. So, good afternoon. Welcome to the session for week 11. So, as you already are aware, this week's session will be majorly centered around the topics of tolerance and autoimmunity and autoimmune disorders. Now, since most of the lectures that were uploaded as part of this week have given you extensive details about the way in which different diseases manifest, I will not be getting into that much detail. What I will instead focus on are the immunological basis of these diseases and what brings it in. I will try to include some different perspectives as per uh, the book that I follow under most circumstances, which is Kobe Immunology, particularly the seventh and eighth edition taken into consideration. And with that, we'll wrap up the entire discussion about tolerance, autoimmunity, the way in which it sets in, what are the different checkpoints that need to be bypassed in order to for the disease to actually manifest, and so on and so forth. So with that in mind, okay, so I have not been able to switch on the video because the weather is horrible here. So my internet is not able to sustain it. If it does work at a later point of time, I will certainly do it. And there will be a lot of background noise, but it's just the weather. So please bear with it. So before we get to the discussion for this week, we'll quickly go over the assignment questions for the past week, where we talked about cytokines and chemokines, the interferon family, the hypersensitivity responses. And if you remember, I had mentioned that we'll be discussing the hypersensitive response in more detail in the upcoming week. In the extra session that we'll be having in the next week, we'll be taking it up in more details then. So for now, let's quickly go over the assignment questions. So the first question was about the GM-CSF subfamily of class 1 cytokine receptors. So the question asked about the roles of the alpha and the beta subunits. What is the exact job of both of these subunits? So the options are that they both induce signal transduction, both are ligand binding, or one of them binds the ligand, the other transduces the signal. Which of the following is true? So let me just quickly go back to that slide so that it will be a little more helpful. So yeah, yeah, I guess this should be good enough. Maybe we can come here. Yeah, so this is the GM-CSF family of receptors. It shows you the schematic representation for this subclass. And if you remember, both the alpha and beta subunits are separately able to assemble, whereby the first condition is where you have only the alpha subunits, where it is assumed to be of low affinity for the ligand, whether it is IL-3, IL-5, or GM-CSF. Whereas when it is forming a secondary complex with the beta subunit, it is able to have much higher affinity for the ligand. And not just that, it is also able to have an extended cytoplasmic extension or intracellular extension sorry, via which the signal can be relayed further. So the beta subunit is the signal transduction subunit, whereas the alpha subunit is majorly associated with ligand binding. Therefore, the correct answer in this case would be option B. That is, alpha subunit binds the ligand and beta subunit transduces the signal. In the second question, we were asked about the redundancy, which is exhibited by IL-3, 5, and GM-CSF cytokines. It occurs because of which of the following reasons? Again, going back to the same schematic, if you see, it is exactly these three ligands that the question is asking about. And the reason that there is redundancy, and if you do not recollect, redundancy is the condition whereby multiple types of signals or multiple receptor ligand interactions eventually lead down to the same or they elicit the same response, or they elicit the same downstream effect. So in this case, you can see that all three of them, I'm not able to get a pointer here, but yeah, okay, I think it should be easy to understand. Once we get into the presentation mode, I'll have the pointer. But yeah, for now, you can see that all three of them are eventually leading to the same downstream signal. And the reason for that is the beta subunit in each case is the same, whereas the alpha subunit is which confers the ligand binding affinity or the specificity is different in each case. So addressing the question once again, the redundancy between them is because the same beta subunit is transducing the same signal, but the alpha subunits carry different specificity. Therefore, the correct answer in this case was option B. Option C and D are of course not correct. That alpha subunit binds the ligand, beta subunit cannot. Both A and C, no. The correct answer is option B in this case. And obviously, you will be able to understand that just from that simple schematic, and of course from the video lectures uploaded for the past week and for any reference book that you are consulting. But if there's any doubt, you can obviously let me know. In question 3, we were asked about the presence of only IL-6 cytokine in the environment induces the development of which of the following? Th1, Th2, Th17, or T follicular helper cells. Again, to address this, let's go back to the schematic. I think I have it here. Yeah. So here we only have Th1 and Th2. Just a second. I'm not sure if I have that exact schematic that I'm looking for. Yeah, maybe we can refer to this particular presentation because I cannot find the schematic. But yeah, the question was asking about IL-6 cytokines and the effect that it induces. So here you can see a different function for IL-6, whereby you can see that it is responsible for acting upon macrophages or endothelial cells. But if we talk about T cell differentiation and the effect upon T cell development, it's not here. I'm very sorry. I don't have the schematic for it here. But basically what it does is it induces the formation of Treg cells. T follicular helper cells, not Treg cells. T follicular helper cells are induced or are driven. The differentiation into T follicular helper cells is driven by the presence of only IL-6 cytokines in the environment. And the reason why this is specifically being mentioned as the only type of cytokine being present is because if you again remember this particular slide, we had discussed how the presence of a single cytokine versus the presence of multiple cytokines can induce different types of effects. Like we had talked about IL-4 being there by itself, which is able to allow the switching of activated, which is able to induce the proliferation or class switching to IgE, either in isolation or in a redundant manner or synergistically with IL-5. But when there is the presence of interferon gamma in the environment, it blocks this class switching, and therefore this step is getting inhibited. So therefore, whether a cytokine is present in isolation or is present along with another cytokine or any other stimulating or antagonizing factor, it will determine what is the downstream effect. So in this case, only IL-6 induces the development of T follicular helper cells or Th cells. The next question was about the signal transduction pathway downstream of these cytokine responses. So the STAT dimer binds to dash element on the DNA, which leads to the expression of interferon-stimulated genes. Fill in the blank. So if you remember the first lecture for the previous week, which was about interferon, which was about cytokines and the signal transduction pathway of JAK-STAT signaling that actually modulates the downstream response, then you remember that, okay, I'll again go back to the figure because I think it should be more helpful. Yeah. So if you remember this exact schematic, so after the cytokine binds and allows receptor dimerization of the JAK receptors, which are present on the surface, there is autophosphorylation, allowing the followed by the phosphorylation of STAT dimers, which act as transcription factors. And therefore, after STAT has been phosphorylated, it dimerizes it, translocates into the nucleus, sits on the correct DNA and regulates or controls its transcription. So the area actually where they are able to sit, the region of DNA where they're able to sit is called as is the response element for that particular interferon, and therefore it is the ISRE or interferon sensitive response element. It means it is able to sense the presence of the cytokine or the interferon and is able to drive the transcription of the downstream gene. So in this case, the correct option was option A, and this is a very direct question from week 1 itself. The other options are simply just made up options whereby they have, modulated the option in such a way that it can be a little confusing. But yes, you just need to keep this in mind that interferon sensitive response element is the correct answer. Coming to week 5, oligoadenylate synthase or OAS activates RNase L by the production of A 2' 5' adenyl cyclase, B 2' 5' adenosine diphosphate, C 2' 5' oligoadenylate, or both A and C. So in this case, it is simply oligoadenylate when it is present. Then oligoadenylate synthase is able to activate RNase L. So that is the correct option. Again, not much to elucidate here. That is the correct option given. Coming to the next question about hypersensitivity. So I don't think I had discussed this myself in the previous session, but it has been already elaborated in the lectures. We will actually take this up in our extra session next week, where I'll be elucidating on each type of hypersensitive response in more detail, along with any questions from the entire course. But for now, let's address the question given. So an Rh negative mother may develop a hypersensitivity reaction if she comes in contact with Rh positive fetal blood during her second pregnancy with an Rh positive fetus. Which type of hypersensitivity response is this? So I think I don't need to elucidate much on it. Each type of hypersensitivity response had an example. Okay, maybe I'll show you the table for just a quick recollection. Yeah, so this is what you see here, the type 1, type 2, type 3, and type 4 types of hypersensitive response. And among these, the condition that it is referring to is actually erythroblastosis fetalis, whereby you can see that an Rh negative mother comes in contact with a positive fetus during her second pregnancy, and that will induce a very severe response, and it can actually be fatal. Sorry. It can actually be fatal in a large number of cases, but there is a current strategy whereby a particular injection, or basically like a vaccine or drug, is given, which is called as ROGAM. So I will be discussing this in that extra session. But for now, if it is interesting to you, you can certainly look it up for your own reference. But yes, like the question has asked, if you look at the example for type 2 hypersensitive response, then you can see that erythroblastosis fetalis is actually an example of the same, and therefore the correct type of hypersensitive response is type 2. I hope it is clear that that was a very, it was just an example based question. They've not even asked you about the details of the disease or the response elicited. It's simply the type of hypersensitive response that is. Now, to be able to tackle these types of questions rather, what you need to do is just, you need to keep in mind that each type of hypersensitive response, one example is clear to you, and you know the mechanistic details of it as well. It might not be necessary for an MCQ-like question always, but it's better to always, have it conceptually clear for your end. So coming to question 7, the release of vasoactive amines is commonly observed in which of the following? IgE-mediated type 1, type 2, type 4, type 2, type 1, type 4, or IgM-mediated type 2 hypersensitivity. Again, here you can see that IgE-mediated hypersensitivity is leading to degranulation, which is, sorry, it's a little zoomed out. Yeah, IgE-mediated type 1 hypersensitivity is leading to the degranulation of mast cells, and it is followed by the release of these vasoactive amines or vasoactive mediators, leading to the actually carrying forward that hypersensitive response. So IgE-mediated type 1 is the correct answer in this case, which is option B. Which of the following events are associated with the release of histamines from the mast cells? First, cross-linking of Ig with allergen, phosphorylation of phospholipase C by protein tyrosine kinase, protein kinase C and calcium ions help in microtubule contraction, and all of the above. So I don't think I had it in my slides, but it was definitely there in the lecture. Let me just quickly go back. Yeah, I didn't include it in my slide because again, it would have been a little redundant. But if you remember the lecture by Professor Ganguly, then you'll have it in mind that after vasoactive amines after histamines have been released, those histamines elicit an entire separate set of downstream responses, starting off with Ig cross-linking and then all of the subsequent steps. Therefore, option D is the correct answer. So if you want, I can show you the schematic, but yeah, you'll find it in any reference book. So I don't think it's very necessary here, but you should certainly go through it for your own understanding. Coming to the last two questions. So which of the following is a T cell mediated hypersensitivity reaction? Type 1, type 2, type 3, or type 4. So it is type 4, the delayed type hypersensitivity. So again, here you can see cell-mediated hypersensitivity, whereby Th1 cells, they release certain cytokines that activate macrophages or TC cells that mediates the direct cellular damage. So TDTH or delayed type hypersensitivity is manifested by T cells. So it is option D, which is for type 4 hypersensitivity. Coming to the last question, antibodies. Oh, this is just the question I answered without even realizing. So yeah, the question was about antibodies injected into an Rh negative mother to counter erythroblastosis fetalis are called A, ROGAM, B, anti-isotypic antibody, anti-idiotypic antibody, or IgE. So ROGAM is the correct answer in this question. So it is a concoction that consists of a mix that helps in countering the symptoms of erythroblastosis fetalis. So that's the correct answer. And if you want to, if you should actually quickly brush up over the differences between isotype, idiotype, and allotypes of antibody, refer to the slides. We have it with very beautiful schematics. So it would just keep you conceptually sound. So with this, we are done with the assignment for the previous week. And now I'll be starting off the discussion for this week's topics, which was week 11. And of course, if you remember, the entire week was centered around the themes of tolerance, autoimmunity, and autoimmune disorders. Let us first understand tolerance. What is the need? What is the concept of it? Why do we need it? And how is it manifested? So just for a quick reference, this again, everything, most of the things, not everything, most of the things are taken from Kubi Immunology. So completely, I have referred to that book throughout the entire length of the course. But yes, certainly you can refer to any book for your understanding. Kubi just gives you a more quick glimpse of every topic that you need to know, because these are very in-depth topics, and the more you study, the more there is to learn. So for now, you can use it as a good reference book, and its PDFs are definitely available online. Okay, so we are trying to understand tolerance and autoimmunity. What is autoimmunity? Autoimmunity, as the definition suggests, is an inappropriate response of the immune system against self-components. So here, auto, of course, means something which is against your own body or own self. Immunity means it's an immune response. Autoimmunity, as we already know, can cause very serious damage to cells and organs, and can definitely result in a number of chronic and acute diseases such as rheumatoid arthritis, multiple sclerosis, lupus, diabetes, etc. So chronic and acute diseases, I hope you already understand the difference between them. So it actually depends upon how long and how intense the response is, that is, or the duration over which that immune response can persist. So in a large number of cases, you will often find an immune response which is relatively short-lived. When I say relatively, I don't mean as short as as an immune response to a pathogen, but rather the fact that the symptoms of that autoimmune response persist for a short period of time versus something which persists very long term, and it's very difficult to recover from such it's difficult to recover for such patients, and therefore that condition persists in them for almost their entire life. So this is what autoimmunity means. Then what is tolerance? Tolerance are mechanisms that reactive lymphocytes, and they are of two types. So under normal circumstances or under physiological conditions, tolerance is in place. Tolerance is maintained within the body so that your body does not elicit an immune response against your own cells, and it is able to keep it safe. And this entire process, or this entire systemic response of tolerance, is mediated at two levels. A as central tolerance, B as peripheral tolerance. Central tolerance, as the name suggests, is something which is operating overall with respect to primary lymphoid organs, bone marrow, thymus, and it will help in the deletion of B or T cells if they possess receptors specific for self-antigens, either in that case or if it binds them with very high affinity. What do we mean by that? If you remember, we will be going into this as well. If you, however, if you still recall, we had discussed that, when the B cell maturation and T cell maturation processes are taking place, then these cells are sensitized to self antigens so that they can recognize and they can maintain the fact that, okay, this is the part of our own body and we are not supposed to um, act against it or react against it. However, it may so happen that these receptors are binding to the antigen. They're able to recognize and bind to the antigen, but they're binding them with an affinity, or the force of that interaction is much stronger than normal. Or when I say normal, I mean it is stronger or higher than the low threshold affinity. So if it is binding it with if the strength of the interaction is above average or is above the threshold, then in such cases, it can induce an immune response, because once it has recognized the antigen, it will not be able to, untwine or not be able to disrupt that interaction. So that is what central tolerance is taking care of. On the other hand, when we talk about peripheral tolerance, it means that it is occurring somewhere in the periphery, majorly with respect to the secondary lymphoid tissues. And what it effectively does is it renders self-reactive lymphocytes that have slipped through the not so perfect central tolerance energetic or inactive. So it is kind of neutralizing those cells that have slipped through the filtering mechanisms of central tolerance. So that's how it works. The limited lifespan of lymphocytes also limits the possibility of damage from self-reactive lymphocytes. So that's another factor to be taken into consideration, that lymphocytes will not persist indefinitely. So the response elicited against them, if they are in circulation with respect to peripheral tolerance, then in such cases, they will eventually be removed, and therefore the type of response it elicits will be more acute in nature, will not be very persistent, and can be, hopefully can be symptomatically managed if it is not aggravating and leading to some other condition. So that is what is in place. Now, how do you actually define the term tolerance? So as the definition has indicated here, let me get the, yeah, just a second. So as the definition here suggests, what does tolerance mean? Tolerance is a state of unresponsiveness to an antigen. So the antigen is there, but against it, we will not, or we are not eliciting an immune response. So that is a state of tolerance. Then comes the difference between an immunogen and a tolerogen. What is an immunogen? Obviously, as the name suggests, any antigen that is inducing or eliciting an immune response. On the other hand, a tolerogen is something that is inducing, it's also an antigen. Antigen is basically any part of a protein, or any, a stretch of, or a portion of an entire protein, or not necessarily a protein, but any molecule within the body which can be recognized by antibodies. Now, if they are getting recognized by antibodies, they are potentially capable of inducing an immune response. On the other hand, if they do the opposite, that instead of inducing an immune response, they are inducing tolerance within the system, then it is a tolerogen. Now, it may so happen that the same antigen can behave as an immunogen or a tolerogen depending upon the context, the environmental condition, and what is the host status. So, depending on that, it can actually either trigger can either maintain tolerance, or it can instead trigger autoimmunity, and therefore can be very harmful for the host system at large. So, that depends upon the context where we are with respect to which we are talking here. The factors that actually promote tolerance include the following. First, being that very high doses of antigens, which means that if a particular antigen is very abundant within the body, then that there is a very high chance that that antigen is actually a part of that host system, and therefore it should not be taken into consideration as an immunogen, and an immune response should not be elicited against it, and instead there should be a tolerance which should be maintained towards that antigen. So high doses actually make a huge difference, and they help in maintenance of tolerance. Second, the persistence of antigens in the host. That means that if such an antigen, not just at high doses, but the fact that if it is persisting within a host body for a very long period of time, then also there is the possibility that it is part of the host system, and therefore a tolerance should be maintained towards that antigen as well. Third, the intravenous or oral introduction. That if there is something which is getting introduced via IV or via consumption, then via the gut system, then there is a large possibility that it is not very harmful to the host body, and therefore there should be a maintenance of tolerance against such an antigen as well. Then comes absence of adjuvants. That means if the antigen, or the hapten rather, is not so large that it is capable of inducing an immune response by itself, which means that an adjuvant for it is not present, then there is a possibility that it is going to induce tolerance against that particular antigen. And finally, for the last factor is that if there are low levels of co-stimulation, which means that it might be so that the antigen is present, but at the same time, there is no supporting signal or co-stimulatory signal accompanied by the presence of that antigen. Then it will not necessarily be recognized as an immunogen and can instead induce tolerance in a large number of cases. In contrast to the initial belief, some self-reactive lymphocytes are actually recirculating in some healthy individuals, but does not inevitably lead to an autoimmune reaction. Treg cells help to maintain this peripheral tolerance. We'll come to it. So here's what you see, a schematic representation of a mature lymphocyte. Two conditions are there. That mature lymphocyte might encounter a tolerogenic antigen or an immunogenic antigen. If it comes across as if it comes in contact with the tolerogenic antigen versus the immunogenic antigen, the response will be completely different. If it is a tolerogenic antigen, then such a mature lymphocyte will be eliminated from circulation or will be energized. When we say eliminated, it means that such a cell, such a B cell, or such a lymphocyte will undergo apoptosis. While on the other hand, if it is, it might be energized, which means that it will achieve a state of functional sort of unresponsiveness, that such a cell is not getting eliminated from the system, but it is not able to induce a response as well. It's just stuck in that sort of middle ground or that state. And as a result, such cells will not be allowed to proliferate. On the other hand, when there's an immunogenic antigen, as you can see here, then such an immunogen is able to interact, or such an antigen is able to interact with its cognate lymphocyte, and such a cell will be allowed to proliferate so that we can quickly quickly facilitate the removal of that immunogenic antigen from the system. We'll look into the central and peripheral mechanisms of tolerance in a little more detail now using these schematic representations. Again, this has been taken from Cobimmunology, I think the 8th edition, if I'm not wrong, 7th or 8th edition. So you can refer to it for your better understanding. So here again, Then you have the lymphoid precursor cell, which is obviously an undifferentiated form of the same cell. There can be a condition whereby immature, not condition, like over the course of time, that lymphoid precursor will form new clones of lymphocytes. It will undergo division and it will form multiple clones. Now, if the clones are getting exposed to self antigens during the course of development, then what happens? What are the different types of responses that can occur? Also, what you see here is that bone marrow or T cells as depicted here, B or T cells as depicted here. So, what are the different types of responses that can be elicited downstream of this lymphoid precursor getting exposed to a self antigen over the course of B cell development? So, first possible response is that there can be a maturation of those clones which are not specific for self antigens in the primary lymphoid organ. So that is obviously the most safe way or the safest way to filter out a type of reaction. That is, if a particular antigen is not if a particular B cell or if a particular lymphocyte does not encounter its cognate antigen in circulation, then such a cell will face no problem and can quickly proceed towards cell division proliferation so as to increase itself in number. On the other hand, when we find that lymphocytes are a population or a subpopulation of lymphocytes are very specific for self antigens present in these generative organs. When we say generative organs, we mean the thymus and the bone marrow. So when there is the presence of this particular any particular self antigen in that primary organ, and then obviously these developing B cells and T cells will encounter this particular antigen, then in such cases such B and T cells need to be completely eliminated without any doubt. They need to be cleared off from the system, and that is by systemic deletion. Finally, for the third part, is that if there is some reactivity, some low reactivity to self antigens, then what happens is that there's maturation of clones that suppress self antigen recognition. So that these are the regulatory cells. So either there will be complete clearance if there is if B cells are completely specific for certain self antigens, or if they are not specific at all, they would be allowed to divide and there is no problem. And the third possible response is that you are suppressing or you are allowing maturation of those clones which are suppressing self antigen recognition. That means they are not directly interacting with the antigen per se, but what they're doing is they are controlling, or a better word for that is regulating. They are regulating the antigen interaction with its or the antigen recognition by its lymphocytes. So they are modulating or controlling it. So this is all part of the central tolerance of the mechanisms of central tolerance present within our immune system. The next type is of peripheral tolerance, which we have already discussed to some extent. So what you can see there is that there's a mature lymphocyte. If it binds to its any XYZ foreign antigen, then there can be activation of effector cells against that foreign antigen. On the other hand, if it binds to self antigen, it will also have those three possible fates. Either it will be completely cleared off by a phenomenon known as apoptosis. Second is cellular energy, or third, it can be regulated or controlled. So these are the three possible types of response. So peripheral tolerance involves, I'm reading the legend here, it involves deleting, rendering anergic or actively suppressed actively suppressing escaped lymphocytes that possess receptor reactive with or specific for self antigens, and this process occurs in the secondary lymphoid organs. Okay, we'll now go through this experiment. Just give me a minute. I'll be back. Sorry for the interruption. Yeah. So, continuing with the theme of tolerance, we discussed about the central and peripheral tolerance. It was understood by an experiment, it was quite a famous experiment conducted by C.C. Goodnow. This was further extended by David Nemes, where he proposed the concept of receptor editing to further this idea. So what do you see here? First, you have a transgenic mouse that expresses HEL as a self-antigen. Second, you have another transgenic mouse that expresses anti-HEL on B cells. So one mouse which has a self-antigen, the other mouse which has antibodies or has B cells which are specific for that particular antigen. When you cross this two, obviously both of these are transgenic mouse. This is how you plan an experiment, that if you want to see this particular condition, you have to engineer these mice in such a way that they express that gene of interest, which will help you to study its effect. So in one case, we have our self-antigen, and in another case, we have the antibody specific for it. When these two mice are crossed, it means when they are allowed to breed, the F1 generation mouse, it expresses both of these proteins. On one hand, it expresses the self-antigen. On the other hand, it possesses B cells that are specific for that HEL. The question is, what exactly happens there? If you look, here is just a schematic representation of the fact that if you could were able to zoom into the bone marrow of such a mouse, then what exactly is happening there? In such a mouse, B cells that are specific for HEL get negatively selected in the marrow. Most B cells that are autoreactive with HEL will be deleted, and some, in very rare cases, can undergo receptor editing. B cells which are not autoreactive will of course be released. So what you understand here is that it expresses this particular or it has these B cells that are specific for our antigen of interest, but what will happen to it is it will get modified or cleared off or edited within the system in one of many different ways. Either it is getting completely deleted, or if it is non-specific, it is allowed to proceed. When we talk about anti-HEL, they get negatively selected. Receptor editing, as the concept was proposed by David Nemes, he said that the antigen-specific V region or the variable region, if for this you need to quickly recollect the way in which B cell specificity, we had studied it in a lot of detail about all the gene segment rearrangements that are present, the way in which gene segment rearrangement is modulated in a way such that it helps you to generate that extensive diversity. If you remember, 10 to the power 5, 10 to the power 7 orders of magnitude in the types of B cell receptors that can be generated, and we had looked at all the mechanisms that operate to facilitate the same. Now, in case of receptor editing, only the V region or the variable region, which gives you that antigen specificity, will be modified. A normal V region gene segment is switched for the autoreactive V gene segment via the process of VDJ recombination. Now, editing occurs most often with the VL region rather than the VH region, because it's light chain variable region would possibly be easier to edit. So that is what happens within the mouse system. The reason why you do such experiments is that everything else that we are studying by ourselves is an in vitro condition where you are studying certain cells. But the response of an individual cell versus that same cell in a system within a living host can be different or can be controlled by many different methods. So in this case, you can see the way in which receptor editing can help in the sustenance of such cells, which might bear specificities for some self-antigens. So this was basically Goodnow's experiment. Peripheral tolerance, it inactivates the self-reactive B cells and T cells that have survived the weeding out process in central lymphoid organs. Again, pretty much what we just saw. For peripheral tolerance, when you have mature lymphocytes which are able to interact with a foreign antigen, then in its response, cell proliferation is carried out, and that immune response is being mediated by these particular B cells or T cells, mature lymphocytes in this case. On the other hand, though, when it is coming across a self-antigens, you can see there is either apoptosis or there is anergy of those lymphocytes that can recognize self-antigens in peripheral tissues. Coming to the Treg cells. So Treg, as the name suggests, they are regulatory cells. They form a unique subset or set of CD4 positive T cells that express a very high level of IL-2R alpha chain. IL-2R alpha is also demarcated as CD25. Again, CD, if you don't remember, stands for cluster of differentiation. It's a classification based on the type of surface antigen which is present, and all these antigens have actually been categorized on the basis of that. So Treg cells are a subset of the CD4 positive T helper cells, and they express this very high level of IL-2R alpha. Okay, and they are generated during negative selection in the thymus. Now, Treg cells express the T cell receptor with intermediate affinity for self-antigens in the thymus. And what they also do is then upregulation of the transcription factor FOXP3. Treg cells are able to induce peripheral tolerance, acting in secondary lymphoid organs and inflammatory sites. The mechanism is not very clear, but of course, production of cytokines, IL-10, TGF-beta, etc. are also implicated. So what is happening here? You see a cross-section of a schematic representation of the thymus. And within the thymus, you can see that when there is low affinity for self-antigen, such a cell is directly going towards towards further maturation. When it has intermediate affinity for the self-antigen, there is upregulation of FOXP5, which is a transcription factor, like we just said, and then it leads to the formation of an anti-reg cells, which suppress the reaction to self-antigens. They become regulatory in nature, and they control the response of other cells to the of the other cells, as in other lymphocytes, to the self antigens, and therefore it is maintaining peripheral tolerance within the host system. The third option is that if it has very high affinity for the self antigen, it cannot be modulated, it cannot be edited, modified, or fine tuned, and instead such a cell should be sent forward for apoptosis or for cell death. Reading the legend here, T regulatory or Treg cells are generated from thymocytes during negative selection in the thymus, and they can inhibit self-reactive T cells in the periphery. During T cell development in the thymus, thymocytes with high affinity for self-antigens, as we just saw, undergo apoptosis. Those with low affinity are positively selected and also released. Thymocytes with intermediate affinity, however, encountered within the thymus, upregulate this transcription factor FOXP3. And they become natural anti-reg cells, get released into the periphery, and they keep self-reactive T cell response in check. So now they become regulators or controllers to control their response. Some issue, just wait a second. Okay, just. Now, for the involvement of anti-reg cells or Treg cells in peripheral tolerance, this was the experimental evidence that was given. So they took non-mice or non-obese diabetic mice. Why? Because they are more prone to develop autoimmune diseases. When such mice are receive an input of normal CD4 positive T cells or T helper cells from a histocompatible donor, then it is delaying the onset of diabetes in such mouse. Further characterization showed that a subset of CD4 positive cells expressing a high level of CD25 was actually responsible. So if you remember, this definition stands true for what is the basis of Treg cells or T regulatory cells. Apoptotic cell death plays an important role in maintaining both central and peripheral tolerance. In both B and T cell engagement by Fas by Fas ligand induces a rapid apoptotic death known as activation-induced cell death, or AICD. Mice carrying inactivating mutations in FAS or FAS ligand are not able to engage the AICD pathway, and hence they develop these autoimmune diseases. Blood-brain barrier sequesters many of these CNS-specific antigens from the central and peripheral tolerance systems. Consequently, when this particular barrier is breached, it can also cause reaction to self-antigens, developing into an autoimmune disease. There is actually a very huge offside to this as well. So I think I have it in one of the upcoming slides. But basically, what you need to understand is that one of the ways in which self-tolerance is maintained is that some antigens, no, they are kept sequestered. What do we mean by sequestered? It means that at the time of immune cell development and maturation, that is when B cells and T cell repertoire is getting generated, at that particular time when they are seeing or they are recognizing the body's own proteins or own antigens, at such a time, there are certain antigens which are not exposed, which are sequestered or hidden, and therefore the immune system is not used to them and recognizes them as foreign. So one of the ways in which this is actually maintained is via the presence of the blood-brain barrier, which actually prevents, or its job is to protect the brain from pathogens or from any disease condition under normal circumstances. So yeah, so when that particular under certain conditions, when that barrier gets breached, then it can actually cause self-antigen reactions, and eventually it will develop into an autoimmune disease as it's expected. Just. Now coming to the next part. So in under the topic of tolerance, we basically understood the definition. We looked at both the types of tolerance mechanisms which are in place, central tolerance and peripheral tolerance, who mediates it, how it is controlled, how cells can undergo different types of responses depending upon how intense or how strong the initial interaction is. That means the affinity that it possesses initially. There can be different fates for different types of cells. Now coming to the second part of today's discussion, which is about autoimmune disorders. Okay, so here it has actually been classified as organ specific or a systemic autoimmune disorder. What do we mean by organ specific? Of course, as the name suggests, the immune response is getting directed towards one particular or a particular set of antigens unique to one organ or one particular gland, so that manifestations are limited, more controlled, and that disease is a lot more manageable. The cells of the target organ might be damaged directly by some humoral or cell-mediated effector mechanisms. Alternatively, it might so happen that antibodies are coming in, they can overstimulate or they can block the normal function of the target organ. On the other hand, we have a second set of systemic diseases. In systemic autoimmune diseases, the response is directed towards a broad range of target antigens, and it involves a number of organs and a number of tissues. These diseases reflect a general defect in immune regulation that results in hyperactive T cells and B cells. Tissue damage is widespread, both from cell-mediated immune responses and from direct cellular damage caused by autoantibodies or by the accumulation of immune complexes. So in systemic diseases, as the name obviously suggests, you don't have one particular organ where your immune response is limited or localized, and it involves a larger number of organs and tissues. So there is an overall loss of control or loss of regulation of immune response that results in very overactive or hyperactive B and T cells. And if the tissue damage, if you monitor it, you will see that it is quite widespread, and therefore it's more difficult to manage as well. And more often than not, it will be considered to be a chronic disease because it persists over a longer period of time. Now coming to autoimmune disorders. So autoimmune disorders, if you look at the overall classification, so to say, you will see this is just a tabulated format for easier understanding, where you have the name of the disease, you have the self antigen, which is triggering that disease, or which is the basis of that disease, and the type of immune response which is manifesting that disease in an individual. Again, this has also been classified into organ specific or systemic types. Among them, let's first go over the organ specific types. So first we have Addison's disease, which is from adrenal cells, where they have auto antibodies against adrenal cells, against some antigens on adrenal cells. Autoimmune hemolytic anemia targets RBC membrane proteins via autoantibodies. Goodpasture syndrome, we'll probably be coming to that. Hopefully, I think I had some slides on it. We'll see that. Goodpasture syndrome is for renal and lung basement membranes, and the response is elicited by autoantibodies. Graves' disease is Graves' disease involves the thyroid stimulating hormone receptor, or TSHR, and in this case, the autoantibody executes a very strange reaction, which is that it is stimulating in nature. Now, this has already been discussed in the video lectures officially uploaded for this course, so you are familiar with it. But we'll also look into Graves' disease in a couple of slides. Then comes Hashimoto's thyroiditis, where again thyroid proteins and cells are targeted, immune response mediated by Th1 cells and autoantibodies. Idiopathic thrombocytopenia purpura targets platelet membrane proteins. What does idiopathic mean if you don't exactly know? So idiopathic basically means that if the definite cause or the causative agent of a particular disease is not very clearly known, or is not very clearly established. So not that the disease is not looked into, maybe it has been studied, but you cannot find the very conclusive evidence for one particular mechanism. So in such cases, it is considered to be an idiopathic disease. And thrombocytopenia means it is targeting platelets, thrombocytes, and there are auto antibodies against platelet membrane proteins. Then comes insulin dependent diabetes mellitus, where you have which targets the pancreatic beta cells, and it is elicited by Th1 and Th1 cells and autoantibodies. Myasthenia gravis is involves patients whereby their acetylcholine receptors are targeted and the autoantibodies are blocking in nature. In contrast to Graves' disease, whereby the autoantibodies against TSHR were stimulating, this one is blocking in nature. So again, this is a good example to keep in mind, because you might actually find it useful at some point of time. One example of each subtype. Myocardial infarction is for the heart, where autoantibodies are working against it. Pernicious anemia involves targeting of the intrinsic factor, gastric parietal cells, again via autoantibodies. And post-streptococcal glomerulonephritis, as the name suggests, it is obviously involves the kidney, and there are antigen antibody complexes which are causing that or are eliciting that immune response. And then finally, you have this spontaneous infertility, which can also occur for sperms. Now, sperms are again a very interesting type of cell to study. The reason being that, when you remember when I talked about these antigens which are sort of sequestered within the body, sperms are actually one such subtype which are also usually kept under sequestered conditions. And there is not usually an exposure of antibodies to these. Depends. It actually depends upon the system that we are talking about. But yeah, many a times it can, therefore, it is more prone to an autoimmune response, and it would it can trigger a disease. When we talk about systemic immune diseases, we have ankylosing spondylitis, which attacks the vertebrae. We have multiple sclerosis attacking the brain or white matter. Rheumatoid arthritis, connective tissue is affected. Scleroderma pigmentosa, which is for sorry, that was zero. Sorry. Scleroderma involves the nuclei, heart, lungs, GI tract, kidney, etc. Sjogren syndrome is for salivary gland, liver, kidney, thyroid. And finally, SLE, the most Among the most elaborately studied autoimmune disorders is for systemic lupus erythematosus, whereby the DNA nucleoprotein, RBC, and platelet membranes are affected or attacked by autoantibodies and by immune complexes. So here are some examples of these diseases. So organ-specific autoimmune disease. Again, the name obviously indicates that it is unique to a single organ or a couple of glands. The cells of the target organ can get damaged directly by cell-mediated or humoral effector mechanisms. One example of which is Hashimoto's thyroiditis, which we just saw a little while ago. So here, ABS means basically antibodies. Antibodies are formed against thyroid proteins like thyroglobulins and thyroid peroxidase, both of which are involved in iodine uptake, hence leading to the enlargement of the thyroid gland, causing goiter. Immune effectors in this case are Th1 cells followed by DTH. And here you see the photographic representation of a normal thyroid gland, which has very clearly defined, clean follicles lined by cuboidal follicular epithelial cells. And on the right, what you see is a very diseased thyroid, which is from a patient with Hashimoto's thyroiditis, where this entire thyroid gland is completely and intensely infiltrated with lymphocytes, which are eliciting the immune response present in this case for thyroid proteins like thyroglobulins and thyroid peroxidase. And therefore, there is a huge inflammatory response and immune response which is elicited, and usually is found to coordinate with the uh, with the enlargement of the thyroid gland, and then that patient is diagnosed with the condition concerned. Then comes the contrast between stimulating antibodies and blocking antibodies, which we just talked about from the list. So when we talk about stimulating antibodies, the type of disease it is usually affiliated with is it is Graves' disease. So Graves' disease has antibodies which are very which they are autoantibodies which are specific to the TSHR or TSH receptor. Um, and it induces unregulated activation of the thyroid, leading to the overproduction of the thyroid hormones. So what exactly happens there? Again, this is something that Professor Shudip had discussed in the video lectures, but just to quickly recap it, if you look here, you see a thyroid cell, whereby you can see that there is a feedback loop which operates in the secretion of thyroid hormone. So the pituitary sitting at the top is usually responsible for for releasing the thyroid stimulating hormone, which binds upon the TSH receptor present on these cells, and then it stimulates the hormone synthesis. When there is sufficient synthesis of these, then regulated production of these thyroid hormones will be able to control a negative feedback response, as a result of which, when there is sufficient thyroid hormone present within the system, then further synthesis will be stopped or will be halted from the pituitary gland by stopping the release of TSH or thyroid stimulating hormone release. On the other hand, in a patient who is diseased or who in a in a Graves' disease patient, you will find antibodies which are specific to this particular TSH receptor, and they sit on top. These autoantibodies they sit on top. They stimulate hormone synthesis, and there is unregulated production of thyroid hormones in such a case. On the other hand, for myasthenia gravis, autoantibodies to acetylcholine receptors on the motor end of these muscles blocks the normal binding of acetylcholine and inhibits their muscle activation. There is progressive weakness of the skeletal muscles in each stage. Systemic diseases, on the other hand, are dealt with differently. So systemic diseases involve immune responses directed towards a broad range of target antigens involving a number of organs and tissues, which leads to widespread damage. Its responsible effector cells or molecules include hyperactive BT cells, you have autoantibodies and immune complexes. SLE or systemic lupus erythematosus has autoantibodies to DNA, RNA, histones, leukocytes, um, RBCs, platelets, clotting factors, anti-nuclear antibodies or ANA. Very sorry. So anti-nuclear antibodies are diagnostic in nature. 10:1 female is to male ratio, which means that females are more prone to this particular disease, especially within the age of 20 to 40 years in women. Characterized by fever, weakness, arthritis, skin rashes, pleurisy, and kidney dysfunction. Involves type 2, type 3 inflammatory damage, elevated C3A, C5A part of that complement response, neutropenia, and vasculitis. So it is actually the broad condition is SLE, systemic lupus erythematosus. But yeah, among them, you can find different manifestations of the disease that can technically be present. Just So what you can see here in this particular little girl's image is the butterfly rash, which is very characteristic of patients who are diagnosed with SLE or systemic lupus erythematosus. But again, what you need to understand is that not every patient will carry the same manifestations of the disease. There can either be a fever. It can be something as simple as, fever and weakness and kidney dysfunction. But yeah, this is a very characteristic symptom for this particular disease. So all autoimmune diseases are characterized by the inappropriate response to self-antigens, and either they will involve the humoral branch or the antibody-mediated response branch of immunity or the cell-mediated branch of immunity. Animal models have implicated the CD4 positive T cell as the primary mediator of autoimmune disease. EAE or experimental autoimmune encephalitis, encephalomyelitis can be induced in rats by injecting them with a particular hapten, which is MBP, or it's an antigen actually, but it's very small, so it's not capable of inducing an immune response. So it is myelin basic protein or MBP, which is injected along with CFA or complete Freund's adjuvant that increases the overall molecular weight and makes it a lot more immunogenic. So this is injected into a normal rat and is believed to induce a condition which is very similar to that of multiple sclerosis. Otherwise, it is exactly known to be experimental autoimmune encephalomyelitis. MBP specific T cell clones can be generated by culturing lymph node cells from these rats with MBP. When these T cells are injected into normal animals, they will, most of them will develop EAE and die, although a few will recover. So what you see, you take this particular hapten along with its adjuvant, which is MPP plus complete Freud's adjuvant, and inject it into a normal rat. Such a rat will be now considered to be a model for experimental autoimmune encephalomyelitis, or EAE, which is a condition where the rat is effectively paralyzed. From such a rat, if you take the lymph node cells and you select for those and you select for MBB specific T cell clones, you can culture them together. And now these specific T cells are injected into a normal rat, then it again induces EAE. So therefore, what you can see is that autoimmune disorders will be then triggered in this manner, and then most of them will actually die. In some rare cases, you will find recovery. Additionally, it has also been shown that the disease can be treated, or rather prevented, by treating animals with anti-CD4 antibodies. An individual who is susceptible to autoimmunity must possess MHC molecules and T cell receptors capable of binding self-antigens, since T cell recognition of the antigen involves a trimolecular complex of TCR, MHC, and antigenic peptide. Further, just give me a moment. Furthermore, the release of sequestered antigens can also induce an autoimmune disease. Any tissue-specific antigens that are sequestered from circulation and therefore are not seen by the developing T cells will not induce self-tolerance. Like I just mentioned, that there are certain locations within the body, like the retina, for example, sperms, for example, or brain antigens, for example, they are sequestered and they are not recognized, and therefore they do not normally it will not induce self-tolerance. And if at any point due to some brain trauma or some sort of an accident, if there is an if these antigens are released into the bloodstream, then there will be an autoimmune response which is induced against. Trauma to tissues following either an accident or a viral infection or a bacterial infection might release some sequestered antigens into circulation. Exposure of mature T cells to such normally sequestered antigens over time might result, at a later time, might result in their activation. So you have some sequestered antigens that are normally present within the body, and there is no harm caused in such cases. However, if under such traumatic conditions, that if there's an accident or some other some other form of exposure, under such conditions, it would lead to the exposure of these sequestered antigens, and therefore there will be an activation and an induction of the autoimmune response. Examples being the release of sperm antigens after a vasectomy, the release of lens protein after eye damage, the release of heart muscle antigens after myocardial infarction have been shown to lead on occasion to the formation of autoantibodies. Data indicates that intrathymic injection of sequestered antigens, intrathymic injection means you are directly introducing the antigen into the thymus, that can often result in the development of tissue-specific autoimmune disease in animal models. So what you do is you introduce your antigen of interest directly into the thymus of that particular of a particular individual, and then it will prevent the development of disease under such conditions. Examples are, first, the prior intrathymic injection of a pancreatic islet beta cells prevented the if you inject the beta cell antigens into the thymus, it will prevent the development of autoimmunity in nod mice. Yeah. MBB prevented EAE in or experimental autoimmune encephalomyelitis in susceptible rats. Yeah, this we finished it. Another way in which autoimmune diseases can often be manifested is via molecular mimicry. What does that mean? Obviously, you understand by the name mimicry that there is a copy of something else. What, in this case, what do we mean by that? It means that there is some molecule which is getting mimicked by something else, leading to an autoimmune disorder. What does it exactly entail? A number of viruses and bacteria possess antigenic determinants that are identical or quite similar in conformation or primary sequence to normal host cell components. Molecular mimicry is therefore probably one such mechanism that leads to autoimmunity. If you look here, you have the human cytomegalovirus IE2 and the HLA-DR molecule. And you can see some overlap or some consensus residues which are common between these two types of antigens. Poliovirus VP2 and the acetylcholine receptor, you can see the common amino acids there as well. Papillomavirus E2 and the insulin receptor, again very similar. Rabies virus glycoprotein and insulin receptor, again very similar. Klebsiella pneumonia nitrogenase HLA-B27 molecule. Adenovirus adenovirus 12E1b and alpha gliadin. Human HIV immunodeficiency virus or HIV P24 and the human IgG constant regions, they are exactly the same if you look at it. If you look at the sequence here, there is complete overlap in the sequence between these two. So that is another way why another reason why HIV is such a difficult condition to completely recover from. Then comes the measles virus P3 and corticotropin, measles virus P3 and myelin basic protein as well. So basically what you see, the human protein is listed second and you can compare it with the pathogen which has that overlap with the host protein under these conditions. Now, when such a pathogen is able to is introduced within the body, then one of two things can happen. That either you will find that such an antigen it is, say for example, if we talk about HIV, it has complete overlap. So in such cases, either its recognition can be very difficult because it will appear like a self antigen. As a result, it will it will execute immune evasion, which means that the immune system will not trigger a response to that as immediately as it will for other antigens. That is one possibility. The other far worse possibility, or just equally worse possibility, is that it recognizes that particular antigen, and the immune response that is elicited against a pathogen like this, a molecular mimic pathogen like this, is also non-specifically or promiscuously targeting the self antigen and therefore triggering the autoimmune condition, which will last for a very long time because there will be a certain number of antigens throughout the whole body, so the response will be triggered. So this is just the mechanism that I wanted to explain about molecular mimicry and how it eventually contributes to autoimmune disorders. Coming to the last slide for today. So this is something you've already read about, which is the autoimmune regulator or AIRE. What is it exactly? I've not gotten into the details of it because you're already quite familiar. Um, just a second. Yeah. So, AIRE is a transcription factor that induces the ectopic transcription of proteins that are normally expressed in a tissue-specific manner in the thymus. It is encoded by the APECED, which is autoimmune polyendocrinopathy, uh, or polyendocrinopathy candidiasis ectodermal dystrophy gene, APECDG. AIRE induces or enhances the clonal deletion of self-reactive T cells in the thymus. It controls the expression or the ectopic expression of a subset of peripheral tissue antigen genes in the thymus. One such gene is preproinsulin 2. So more details about its function you've already come across in the official presentation uploaded by Professor Shudeep, and of course you can refer to any book if you want to study this in more detail. I did not get into it because I think it was the first topic introduced for this week, and he went into a lot of depth. Professor Shudeep went into a lot of depth about this topic, so I did not went did not venture into it as extensively. So with this, we come to an end for today's presentation. I will go through a very few questions, previous year questions for easier understanding. Just give me a minute. Okay, so the first question is about the term xenoimmunity. What does it exactly refer to? First, the immune response to a foreign antigen, immune response to a self antigen, immune response to transplanted organs, or immune response to commensal microbiota. So it is actually when we talk about xenoimmunity, it talks about something which is present, which is in concordance with what is already in the body. So it is immune response to commensal microbiota, which are under normal circumstances present in a commensalistic relationship or a type of symbiotic relationship, let's call it that, involving some resident microbiota, and a response elicited against it is considered to be xenoimmunogenic or xenoimmunity under the purview of xenoimmunity. The principal mechanisms of peripheral tolerance include A. Suppression by Treg cells, B. Energy, C. Activation induced cell death, which is AICD, or D. All of the above. So we have discussed this in using multiple schematics that if you talk about peripheral tolerance, any one of these three methods can actually come into play there. First, there can be complete energy or suppression of any functional response. Second, it can lead to Treg mediated suppression. So there is a suppression of self intolerance. And third, for activation induced cell death or AICD is of course certainly there. So the correct option is option D in such a question. So you basically look at each question, the options of each question, and consider them to be the correct answers for a minute. Look into the possible effect of that correct answer and then narrow down. So in this case, it is all of the above or option B. The function of autoimmune regulator or AIRE gene is to do which of the following? So I just showed this in the last slide. But yeah, let's go over the options as well. First is to inhibit T cell development, to aid or help T cell proliferation, to promote the expression of tissue specific antigens for T cells to recognize self. That is actually what is the job of AIRE or none of the above. So the correct answer in this case is option C. That is to promote the expression of tissue specific antigens for T cells for self recognition. Question 4 is about myasthenia gravis. What is it caused by? It is caused by the production of autoantibodies against myelin basic protein, TSH receptor, acetylcholine receptor, or none of the above. So in this case, it is ACH or acetylcholine receptor against the acetylcholine receptor, basically inhibitory or blocking in nature. In SLE, which of the following receptors is involved in activating autoreactive B cells? I don't think I went into details about it, but it is actually TLR9. Yeah, I did not go into details about it. It was probably part of the lectures. But for your own reference, for your own understanding, you can read about this in more detail. But it is TLR9 that leads to the activation of these autoreactive B cells. Sixth is about inhibitory cytokines. They are produced by natural Treg cells or nTreg cells to inhibit self-reactive T cells, IL-10, TGF-beta, both A and B, or none of the above. So it is option C that inhibitory cytokines are produced by these nTreg cells to inhibit self-reactive T cells involving both IL-10 and TGF-beta molecules. A syngeneic graft is tolerated, as it involves a transplantation between people who are genetically similar or identical. So the options here are genetically distinct, identical, same individual, or genetically distinct individuals for MHC. So syngenic graft will be obtained from a genetically identical individual. From distinct individuals, there might be intolerance. Genetically distinct individuals, especially for the MHC, there will definitely be intolerance, and same individual will be auto transplantation. Question 8 is about major histocompatibility proteins. What are they actually responsible for graft rejection? And the reason for that is A, antigenicity, secondary structure, polymorphicity, or helical propensity. So graft rejection is majorly due to polymorphicity. Finally, for the last two questions, so indirect allo recognition is mediated by the presentation of donor molecules to the T cells by indirect. When we talk about indirect allo recognition, that is because donor molecule is being presented by self-MHC molecules. So that is leading to allo recognition in an indirect manner. So it is option B. Finally, the fetus is an allograft that is not rejected because of which of the following factors. Again, this was extensively discussed in the video lectures by Professor Shrudeep. So, first is the absence of MHC2 on the placenta trophoblast layer, nutrient depletion by action of the IDO enzyme, upregulation of induced Treg cells, or all of the above. So, it is actually all of these options or all of these phenomena which altogether lead to prevent the rejection of the fetus, despite it being technically being an allograft. So, with this, we come to an end for today's presentation. To quickly go over the idea for this week, we looked at all of the different topics covered under the purview which is determined by the two professors. So we looked at tolerance, the phenomena, how it participants in that process. We talked about autoimmunity, some examples, some very key examples. Obviously there is indefinitely more to explore in this condition, but basically what we looked at was just some very classical examples that will be um, helpful. And finally, we looked at some questions which address the basic idea we have conveyed. And with this, I think maybe I'll conclude my session for today since there is nobody else here as of now. So I hope these sessions have been helpful. I understand it has been quite irregular for the past couple of weeks, and I'm extremely apologetic about that. It was due to some personal matters for which I was not able to carry out these sessions at a regular time, and I had to keep shifting them. But nonetheless, I hope the uploaded videos are at least helpful for people and you're able to go through it, or at least through the slides for your own understanding. And yeah, I think that's where I'll end the class for today. Thank you so much for joining and all the previous sessions that you have joined and for listening to the lectures because I hope it has been helpful to you. And I sincerely hope that people are able to join the last extra session so that we can have at least a good interaction that day. And with this, I'll end the session for today. For everybody enjoying the holidays, hoping you have a wonderful time. Thank you and bye bye.